it's really my great pleasure to introduce our last speaker until the summer break. Xuesong Li, he's now a postdoc with Harry Croft here, but he got his bachelor's at Zhejiang University in optical engineering, and then went to Hong Kong University to get his PhD in the Hong Kong University of Science and Technology, where he did integrated stimulated Raman, so SRS microscopy and 2-photon film microscopy. So we're super excited to hear your talk on super resolution bioimaging. Thank you so much.
Okay, great. Thanks, Brian, for the introduction. So hi, everyone, I'm Xuesong Li, and I'm currently a postdoc in Harry Croft lab. And perhaps you all already know my supervisor already. And today I'm honored to introduce my previous project finished at NIH before Harry and I moved to Janelia, like maybe last year, like after June and September. And so in this talk, first I will introduce the background and the motivation of why we want to do this project, including the background like the multi-scale biological imaging. Then I will talk about super resolution imaging modality named 3D SIM. So some of you may have already used it. Some of you may have not heard about it, but it's one of the three big, super resolution microscopes in this world, named single molecule, including Storm and the satellite like MiniFlux and STAT nowadays. And also this last one is a structural illumination. Actually, this technology was invented by Matt Gustafson at Janelia here. So it's quite a coincidence that I talk about, our ongoing work following Matt, what Matt have been done at Janelia. And unfortunately, Matt passed away like 10 years ago, but his work is still, continuing and increasing and growing in this field. So in the end, I will present the detailed work on how we improved the actual resolution of 3D SIM, or you can see Z resolution, which is quite difficult in this field. Finally, I will talk about something, what we are going to do, and actually what I'm doing now, which is named as multi-view line confocal super resolution microscope. So here we go. So as we know that fluorescence microscopy is actually an essential tool for biological research nowadays, and there are a variety of microscopy techniques with different application scopes, as you can see here, mainly in terms of the temporal resolution, the spatial resolution, and the imaging depth. So for example, super resolution technology nowadays like STAT, Helm, and the Storm, same MiniFlux, now has enabled resolution so that nanoscale protein distribution could be visualized at very fast speed in living samples. And light sheet microscopy nowadays enables gentle and long-term, very gentle imaging of cellular dynamics in very transparent tissues. And the multi-photon microscopy, including 2-photon and nowadays 3-photon microscopy, can achieve deeper penetration depth, which is longer than 100 micron to 1 millimeters in highly scattered samples. So modern fluorescence microscopy has been developed to cover the entire range of these diverse spatial temporal scales. On the other hand, however, we know that biological phenomena also happens on diverse scales. For example, this phenomenon could occur, cover the entire range of the nanometer scale to meter scale, and over time scales ranging from milliseconds to even days. Therefore, for different biological phenomena, it is quite important to use and choose the right microscopy technique. Although there are so many imaging modalities, right? So one challenge always exists is that the quality of fluorescence imaging is always determined by 4 important aspects or the factors: the health of the biological samples, which means that you can bleach or do photo damage to it, right? And the spatial resolution, that is the meaning of super resolution nowadays. And the temporal resolution, which you can do really fast using resonance scanner or light sheet microscope. And the signal to noise ratio. You definitely do not want to see something which is totally blurred or full of artifacts. So these 4 factors, they are known as the pyramid of frustration in fluorescence microscope, which means that no single factor could be improved without compromising the other 2 or 3. So among all the microscope modalities, actually SAME has both the high spatial temporal resolution and the fast imaging speed. Therefore, I chose SAME in this study as my imaging weapon. So what is SAME? The basic concept behind SAME is actually is that if we collect enough, images with patterned illumination, like here, which is a grating pattern with different orientations and angles, and you collect enough information from the raw data, you can extract the high frequency components from it and do the linear reconstruction to form the final super resolution images. So there are 2 pioneers in this field. One I have mentioned is Matt. The other one is Rina Hanizma in Europe. So compared with the wide field and the confocal microscope, structured illumination microscopy has a double resolution, which is actually not as good as PALM and STORM, but it has some unique advantages that there is no dye or fluorophore restriction with SAME, which means that you can use basically any kinds of fluorophores or fluorescent proteins to do the super resolution imaging without photo bleaching, without photo blinking capability or photo activation. So it's quite different from the single molecule and STAT. And when SAME was first invented, it actually has a relatively low imaging speed, like only 10 Hertz, but already fast enough. I would say that nowadays the SAME imaging speed of SAME has been improved quite a lot, which can go like over 100 frame rate. And so it is really a good choice for lifestyle super resolution imaging. Based on the different illumination patterns, as you can see here, actually there are 3 different basic variants of structured illumination microscope. In turf SAME, as shown here, the imaging depth is actually limited to 200 nanometer, which is very close to the curve slip, which means that you can only see something which is close to at the bottom of the cell, and you cannot go deeper. But the resolution can go like 100 nanometers or something like that. In 2D SAME, that is the most commonly used one, or commonly built by some optical labs. The actual resolution is actually the same as a confocal, maybe it's worse. Actually, the same as a wide field microscope, because there is only optical sectioning, but there is no optical super resolution along the z-axis. So the actual resolution is limited to 300 nanometers here, which is same as the wide field. But most people will say that, okay, it's fine for cell imaging because I don't care about Z. And the last one is actually the quite, most interesting one is named as 3D SAME. So 3D SAME actually, why it's different? Because compared with 2D SAME and turf SAME, it has 3 beam interference at the back focal plane here, right? And so it provides not only the lateral super resolution, which is same as 2D SAME is 120 nanometers, but also improved the actual resolution twice. So among all the SAME variants, I would say 3D SAME is the most intriguing one. And not so many labs are actually still exploring 3D SAME. Most of them are still exploring 2D SAME. I would say 3D SAME is quite useful, not only because it has the optical sectioning, but also it has the super resolution capability along the z-axis. However, as you can see here, the 360 nanometer actual super resolution is still worse than the 120 nanometer lateral resolution, right? which means that 3D SIM leads to an anisotropic super resolution microscope. The fine features can be clearly differentiated along the XY view, but still quietly, blurred along the Z view. So here the blurred, I mean, compared with the XY view. So we know that actually most biological phenomena, they are not a plane. They are not 2D, but they are stereo, right? So, and most of their behavior also happens in 3D or even 4D. So here the 4D means that you have a time-lapse 3D imaging. So what makes me feel very interesting is that, so how we can push the actual resolution further or even make it isotropic on the 3D SIM microscope. That is the main target of my research. So let's go back to the wide field. In wide field microscope, the sample is illuminated by one beam only, which is, everyone knows that. In 3D SIM, sample is illuminated by 3-beam interference, right, at back focal plane. So we usually name this one and this one as ±1 order of the beam. And the central beam, some people call it 0th order, some other name it as DC order. So it's the same meaning here. To further improve the actual resolution of 3D SIM, we actually think about a very simple but straightforward method, is to put a mirror on top of it. Then our idea is to introduce 4-beam interference at the sample imaging plane. We should that placing a mirror directly opposite the sample-enabled 4-beam interference, named 4-beam SIM. So we first characterized the 4-beam SIM imaging capability by imaging 100 nanometer yellow-green beads here, which is quite standard for the PSF measurement. And from the XZ view, so here is all the XZ view from the wide field, 3D SIM, and the 4-beam SIM. We can already know that our 4-beam SIM makes the beads look significantly smaller along the X direction. If we zoom in and plot the intensity profile of the central line, we will know that the progressive actual resolution improvement from the wide field, 3D SIM, and 4-beam SIM can be clearly illustrated. And the statistics analysis of the full width at half maximum shows that the 3D SIM doubled the actual resolution from 600 nanometers to 350 nanometers, which is quite consistent with previous work. While our 4-beam SIM further improved the actual resolution from 350 to 160 nanometers. But as expected that both 3D SIM and 4-beam SIM, they maintained a double super resolution compared with wide field imaging along the XY view. So we can see that our 4-beam SIM realized, near-isotropic super resolution imaging with 100 nanometer lateral actual resolution and 160 nanometer actual resolution, or Z resolution. So next, we want to test by it works on beads. How's the performance on the real biological samples? And first, we tested out the performance of our 4-beam SIM compared with 3D SIM and wide field on bacteria, which is small enough to differentiate the fine structures. So we first imaged the live bacteria stained with membrane dye. From the two different field of views here, field of view 1 and field of view 2, you can notice the wide field can barely, resolve cell membranes along the actual views, but could be better visualized in 3D SIM. However, 4-beam SIM provides even greater visual clarity. And this resolution improvement could also be demonstrated and quantified by the apparent cell membrane thickness. But I have to say that that is not the real thickness of the bacterial membrane, because it should be much thinner than dozens of nanometers here. But we can still observe that the improvement is greatly increased, right? And also in 3D SIM, there is a phenomenon in the membrane of the bacteria named invaginations, which is some convex or concave features, which you can hardly differentiate it or observe in 3D scene, but you can quite clearly observe here. So together with the movies I shown here, so here is a Z view, so X view, Z view again. So compare with our 4-beam thing. They all demonstrate that the ability of the 4-beam thing to reveal the actual details otherwise obscured by the diffraction. Next, we verified the resolution enhancement on live bacteria, labeled with the so-called D4A GFP. And D4A is actually known as a protein to assemble at a nascent division size with kind of double ring structures. And although the both 3D same and 4-beam same, they can differentiate these double ring structures, but only our 4-beam same, can provide clear actual views of the clarity of the circularly shaped rings. But if you use the 3D same, you will find that it is something like the ovoid appearance, which is not correct because the Z resolution is not high enough. And after the 4-beam same illumination, you will find that, okay, it's totally a circular ring. It's not a ovoid regions. And 4-beam same could also be extended and used with dual color imaging. So here is a sporulating bacteria double labeled with SPOVM GFP, and the cell membrane dies. So SPOVM is a peripheral membrane protein and is a key component at in the spawn coat. And the dual color 4-beam thing enabled crisp visualization of the SPOVM within the membrane of the mother cell, but such detail was totally lost in wide field imaging. So I can show you the difference comparison here. So there is no 3D same comparison, but it's something, the performance will be something between the 4-beam same and wide field. So let's talk about something about U2S cell called 7 cell, which is also quite usually used in same imaging. So next we examine these U2OS cells with larger field of view. So this fixed U2OS cell immunolabeled with TOM20 GFP. Sorry, it's TOM20 antibody staining here. So it marks as the outer mitochondrial membrane. So when performing the wide field imaging, diffraction actually obscured the inner mitochondrial space. So you can see nothing inside the mitochondria, but we know that we only label the outer mitochondrial membrane. It should be something empty inside. But by contrast, the lateral view produced by 3D same and 4-beam same could easily distinguish these void regions. In actual views, however, only 4-beam same could effectively resolve these void regions, and the relatively worse, let's say 350 or 360 nanometer actual resolution of 3D same still blurs the actual view, which makes you think that, okay, there is no void region inside it, or it's hard to say, but it's quite clearly visualized here. Next, we stained the leading U2S cell with mitochondrial green, marking actually not the outer mitochondrial space, but the inner mitochondrial space. So the top image is the lateral view, and the bottom image as the actual view corresponding to the 3 dashed lines shown here. If you look at the movie, both the lateral view, here's the lateral view from the different Z depths, and the actual view is shown here with different position of the Y. They all have very high resolution to discern the inner mitochondrial structures. Okay, then we also imaged the cytoskeleton structure composed of microtubules and the vimentin filaments. So actually, we instead of the colocalization, we observed an apparent so-called stratification phenomenon, which means that the microtubules and vimentin, they are not like overlapped with each other, but one layer by the other layer along the z direction. And we can also observe examples in which local enrichments of the vimentin is appeared to fill in the gaps between the microtubules. And our observation is also quite consistent with a recent study showing that vimentin filaments somehow can template the cell structure to stabilize the cell polarity during the cell migration. Finally, we imaged the phyloidin labeled actin filaments and dyes in the membranes in fixed mouse liver cells. So here's the actual views from this site. Revealed actin appears to be encapsulated within the fine membrane structures, likely the photopodia, and the small actin enrichments is shown here, and pore-like structures like here, something like a hole here, was also found within the layered regions, these small regions. So I have to say that despite these successes of the 4-beam SIM compared with wide field and 3D SIM, it's not perfect. One important reason is that the volumetric time-lapse imaging with 4-beam SIM is quite challenging. For example, even if we're using the bright and the photostable dye provided by Luke's Levy's lab, which named as Potomac Gold, and also tried some other GFK dyes here, but with only 10, maybe 10 volumes, 10 points, you will find the phototoxicity and the photo damage. There is no bleaching, but laser, but due to the photo damage, it's not due to bleaching. This is actually not surprising because you have to think about the image formation algorithm behind the same, that you need to acquire enough data sets to do the reconstruction. For example, this cell, we take only 4 micron thickness, and you need to take 67 actual steps, because one step you need to acquire like 125 nanometers, which is quite fine. And each layer you need to take 3 directions with 5 different phases, which is 15 images. So if you multiply them together, you find, oh, oops, I need to acquire like more than 1,000 images, raw data, to do the final reconstruction. That is really difficult, to make it difficult to do the 4D time-lapse imaging. In fact, bleaching is always a big problem in lifestyle imaging. There is a very classic paradox. I have to say that we need to use high laser power to get high SNR images, right? Or you extend the to longer exposure time. But this also implies that you need to get if you use longer exposure time and high laser power to get high SNR images, you will get high bleaching, and this in turn prevents the time lapse for the high resolution imaging. As one video shown here, this is due to a cell transfected with metal tracker, metal GFP here, which is not acquired by 3D SIM or 4-beam SIM, but acquired by a conventional instant SIM technology also developed in Harris lab. So one layer is only exposed one time. Even this, you will get quickly, rapidly bleached images with time. So we want to ask a question. So how to perform the volumetric time lapse, or say 4D imaging in living cells? Actually, we have 2 solutions. The first one is to turn down the illumination power at the cost of signal to noise ratio. The second solution is to use 3D SIM, which is like 125 nanometers per step instead of the 60 nanometers per step for 4-beam SIM. Then you can save half the time and also decrease the laser usage by half. So we decompose the overall challenge into 2 sub problems. The first question is how to denoise 3D SIM images with low SNR, The second question is how to improve the actual resolution based on the 3D same data without adding the mirror. We then considered about very popular, computational microscopy nowadays using deep learning to improve the actual resolution of the 3D same without introducing additional laser dosage of the laser power. So first, I want to talk about some more recent work in this area, which is using deep learning for image restoration, deep learning, and to increase SNR. So if we look back at the pyramid of frustration, as we talked about, so there are only 3 factors here. It's a little bit different from the one I put on the the first slide, which has also has a sample health. If you look at the pyramid of frustration, there are actually trade-offs in the physics of microscopes. So deep learning, however, uses a data-driven prior knowledge to recover, principled way to recover what is possible. So in recent years, many different neural networks has been developed and used in microscopy. I won't talk about too much details about this. So I will say that CARE is the most important one in this field. And the DDN and Richard Lucy network is also utilized and invented in Harris lab here. And the RCAN by another, previously was also postdoc at Janelia named Ji Chen, currently working at NIH AIM. So he used and modified the RCAN from 2D to 3D to do the denoising, which is also quite successful. And my PhD senior lab mates during my PhD period, named Dong Li, now previously is also postdoc in Eric Bezik's lab here. So he invented the DFCAN and DFGAN in this field. And more recently, he named this as a regionalized deep learning. So it's all like different kinds of imaging neural networks in this field to improve the progress. So broadly speaking, the workflow of all of this methods is quite similar. It's not magic, but it's more like a black box. So in the case of denoising, you need to acquire groups of low SNR data and the corresponding high SNR data and input them to the neural network to train the neural network, right? Then you can challenge this neural network with some low SNR data you have never seen before. It shouldn't be inside the low SNR data training data. It's something as an input data or test data you have never used before. Then you can restore the images with high SNR. So our group collaborated at NIH at that time, collaborated with S-Vision to use the so-called 3-dimensional residual channel attention network, or you can say 3D RCAN, to produce better images with higher SNR on different microscope data sets. So actually, the real strength of these neural networks is that you can apply them not to, for the training data is always based on some fixed and dead cells, because you definitely do not want, when you acquire the training data, there is some registration issue, right? When you acquire the low SNR and high SNR, but you can train, you can apply this model, the live cell imaging, right? And most microscope, super resolution microscopes, actually, they were illuminated throughout the samples. So they are not light sheet type microscopes. So bleaching is a quite serious problem. But if the laser power is low enough, the photo bleaching rate is small relative to the fluorescent protein synthesis rate. Then you can, theoretically, you can do imaging with unlimited time. So in the 2 color sample here, where the mitochondria in green and the lysosome in red, the lysosome data is actually barely visible as the cytoplasma here. But after 3D RCAN denoising, we could restore the data to the point that we could quantify the mitochondria and the lysosome interactions over time with over hundreds of time points. This was quite amazing. The first time I saw this video when Gigi, produced this data, because this is something we have never seen on any super resolution microscope before. So I used the similar concept also using a 3D RCAN to denoise my low SNR 3D same data. It's not based on full beam thing, but it's just 3D thing. You can notice that the progressive contrast from the raw 3D sim reconstruction and after the first denoising network and after the second denoising network. So here is a little bit different from our previous work is that I utilized the two-step denoising. The reason why 3D sim can be used with two-step denoising is that we have the raw reconstruction, you have the raw data. So if you build two neural networks based on the raw data and based on the raw reconstruction, we can do this two-step denoising. So it's a little bit complex to explain, but I can show you later. And we also, not only want to improve the SNR, but also want to improve the actual resolution to make it isotropic. Deep learning could also do this for resolution enhancement, as shown here. So it's also instant sim modality. It's not 3D sim or full beam sim. If you look at the microtubule network in the living T cells, this is input data. You can see the actual view is just okay. You can still discern something, but the RCAM prediction cleans up and the actual view and the deep blurs the overall structure, particularly in Z. So if you look at the actual cross-section of the mitochondria sample here in the raw data, right? It is really hard to differentiate any mitochondria, but after 3D RCAM prediction, it is easier to do so. But the overall, resolution improvement is not, just okay. It's not as satisfied as what I want. So we wonder whether there is a more efficient way to improve the actual resolution of 3D sim reconstruction data or the raw data, and even make it isotropic. So here is my imaging modality. We propose a new deep learning actual view from the actual view itself. So previous work, when they improve the actual direction resolution or say the Z resolution direction, they will usually use the XY view as a training model. But we know that XY view will be quite different from XZ view. A simple explanation would be that gravity, right? So if you look at the cell, the cross section will be totally different from XZ and XY. So here, I invented a relatively new strategy. It is not a new model. It is not a new neural network modality or say a new neural network, but a different way to use it. So here we can observe a relatively clear visualization of microtubules in the 3D projection movie, especially with a crisp actual view. But the question is, can we trust the prediction results, based on this method? The answer is, luckily we have the full beam thing as previously invented, right? Then we can use the full beam thing with near-isotropic super resolution to validate, right, the deep learning prediction is correct or close to correct or not. So we first compare the same microtubule sample produced by 3D sim, which has 360 nanometer actual resolution, full beam sim, which has 160 near-isotropic actual super resolution, and deep learning prediction. Although all the three methods offer the similar lateral super resolution here, which is all 120 nanometers, so here is OTF, or you can say something like the in the frequency domain to explain the resolution. Along the actual direction, however, both full beam sim and deep learning prediction, they show close visual, and quantitative, agreement to differentiate the relatively peaks along the actual view. So here, I want to emphasize that the value of super resolution microscopy and the deep learning to improve or deep blur the actual view is actually not to observe the smallest objects as small as possible. So if you can observe some 10 nanometer beads, does not mean that your resolution is 10 nanometers, right? But if you could differentiate the two beads with the distance with 100 nanometers, and we can say, okay, your actual resolution, super resolution capability has already reached 100 nanometers. So next, we also performed the two-color imaging of caveolin 1 and cavin 1, two components of caveolar codes in the cells. Although most of them, marked a distinct position inside the cells, but we could still observe a very small region with co-localized signals. If we look at the deep learning prediction of the caveolar one, it's actually quite interesting because it appeared to resolve ring-like structures. That is a 3D sim input. That is deep learning, and some semi-partial or say partial ring structures, right? And some spherical puncta or say dot-like structures. So okay, we now have the denoising model and the actual resolution enhancement model. And these two models are specifically designed for 3D sim. And the high quality 3D sim prediction produced by the two-step denoising model could then be fed into our actual resolution enhancement model. Then we could turn down the illumination power, intensity, and perform 4D isotropic super resolution imaging on living samples. So here we perform 3D sim imaging of TOM20 GFP in live U2OS cells over 50 volumes time points without substantial photobleaching or obvious phototoxicity. And you can notice the progressive contrast and resolution. So here is actual resolution. You can see the void regions inside, along the axis view, and the resolution improvement along, that is a raw reconstruction without any manipulation, which means that the SNR is low and the resolution along the axis Z view is terrible. And after the first step, denoising, okay, looks a little bit better, but the actual resolution is still not good. After the second denoising and after the isotropic resolution improvement. It's just another movie, another example to show the, we can also image the lysosome dynamics with LAMP1 GFP in live U2OS cells. The similar concept is here that after the first denoising, second denoising, and the third isotropic second step, last step isotropization method, or the actual resolution improvement. Of course, deep learning could also be used for dual color live cell imaging, right? We also always talk about dual color. And so this U2OS cell is actually expressing LAMP1 GFP, which labels lysosomal memories, outer memories. And it was additionally labeled with lateral tracker red, which labels lysosomal interior. So we can say the volumetric imaging over 60 time points shows the potential of this multi-step deep learning method. So it's all based on some quite standard. The previous movie is all based on some quite standard, live cell imaging with TOM20 GFP or metal tracker red, something like that. Finally, I want to say, this one could also be used to investigate some basic or say some phenomenon which has already been investigated before, but not at high resolution in biological research. And here we investigated the microtubule dynamics in a live Jurkat T cells. So this Jurkat T cell has already been activated on CD3 coded antibody. And the deep learning prediction revealed the finer details right here. And the small filaments, compared with the raw 3D sim reconstruction, which is just terrible. And over 100 time points, we actually can observe a phenomenon so-called the movement of the two microtubules and the merging together, encycling, and the final, the living together. And this system could also be used to investigate the spatial relationship between the so-called MTOC, or you can say it's just like the centrosome, right? The centrosome position between the between the centrosome position and one of the segmented filaments microtubules. As we can see in the movies, MTOC repositioning is closely related to one of the segmented microtubules movement. To summarize this study, mostly finished at NIH over the past 4 years, we want to say that we demonstrated two practical but complementary methods to improve the actual resolution based on the 3D SEM microscope. Okay, the first is a so-called 4-beam SEM, which is a totally purely physical-based method, and it needs no information about the sample. But the phototoxicity and the photobleaching is really a challenging problem. The second method we're using here is based on deep learning. So there is actually no hardware limitation. You don't need to modify your 3D SEM system, what I need. But the question is, you need to acquire like over 50 different data sets of low SR and high SR data to establish the neural network for the reconstruction. However, I have to say that the 4-beam SEM and our multi-step deep learning is actually, they are not isolated, right? They're actually closely correlated with each other, because we can combine these two methods together and to reduce the phototoxicity and improve the potential for 4-beam SEM using the deep learning pipeline here. And conversely, we can also use the our 4-beam SEM to validate the multi-step deep learning result is correct or not. And our deep learning pipeline could also be used to improve the actual resolution potentially of other imaging modalities. Actually, we have already tried confocal and STAT, and our instant SEM it works not as good as 3D SEM, but indeed improves the actual resolution a bit. So in the last part of this talk, I want to discuss about it's actually not future research plan, but it's actually what I'm doing now at Janelia, since Harry and our lab moved here. So in general, our future work still includes two parts. The first one is optics part, second one is algorithm part. So as I mentioned, that the image depth of 3D SEM and 4-beam SEM, whatever SEM, right? Whatever conventional super resolution SEM we have nowadays, the imaging depth is limited to 20, at most 20 micron. Why? Because if you go deeper, the interference pattern will be destroyed. It's based on interference. The tissue scattering will be a problem. So if the 4-beam SEM or 3D SEM, they were forced to image something that is not suitable for, you will find that the image looks just garbage, it's full of artifacts. So it is the adult worm, C. elegans, try to be imaged on 3D SEM, looks terrible. But we know that biological samples are more than just single cells, right? So I want to say that for worm, flies, fish, mice, so are there any more suitable super resolution modality or say microscopes that can be used to image them? First is at least 50 micron or even more imaging depth, because after all, it's still super resolution. We are using now still based on single photon. You cannot say, oh, let's say 200 micron is too crazy for single photon. It's not 2 photon, right? We also want to maintain the high resolution, spatial resolution, and also the high temporal resolution. So more recently, actually 2 years ago, our staff scientist at NIH NIBIB named Xuesong Li, and one exchange, very talented exchange PhD at that time as a PhD student. Now I think she had already graduated from Tsinghua University. So they too collaborated with ASI to invented this very special multi-view confocal microscope system. As we know that in a single view confocal microscope, let's say an inverted one, right? The resolution is terrible in Z, but it's just barely good along XY. So it's actually the same as 3D SEM, which is an anisotropic super resolution. It's not as super resolution. It's just anisotropic resolution, right? So the advantage of this triple view or say multi-view confocal system is that you can combine the 3 views and using the registration and deconvolution, to make it the isotropic. And although it's isotropic, but it's not super resolution, as demonstrated here, it's sub 300 nanometers. Oops, this one. I think should be some movie here. Let me see. Yes. Anyway, so there is a movie. Another advantage of the triple view system is that you can actually compensate from the bottom view to make the penetration depth even longer to cover the whole worm of this of the C. elegans in the adult whole worm. You could count the nucleus more accurately at a deeper penetration depth, which is over 50 microns. But that is almost impossible with a conventional, let's say, area scan or a conventional inverted confocal microscope, because it can only image half of the worm, and if you go deeper, scattering will dominate. So another feature of this line confocal design is that we actually can do structural illumination or say super resolution on this confocal base. So if you look at these patterns here, if you like these patterns here, they were actually not generated by SLM. They were power manipulated by AOTF to give different patterns with different time, right? Then you will have something pattern close to a grading projection or SLM projection, based same structure, but it is intensity illuminated. So we can modulate the image by turn on and turn off the illumination at very high speed during one image. Then we need to acquire 5 different phases, or you can say 5 different images to do the reconstruction. So here the reconstruction is not based on linear reconstruction, but based on photo reassignment, which is kind of a variant of another structural illumination technique. But you know that you need to still like the 3D SIM, you need to 15 images, 2D SIM, you need 9 images. Here, line confocal SIM, you also need 5 images to do the reconstruction. So consider about what we have done before. We think there are still many aspects we can, drawbacks we can overcome, or say some, shortcomings we can improve, right? So several factors motivate us to further improve the performance of this line confocal, or say a triple view line confocal system. The first one is that we must change this modular design, because here we look at this. It still looks something quite similar to dice beam, but I have to say that it's not a light sheet. It's a confocal, so the excitation objective and the emission objective serve the same view, serve the same view, not serve the 2 different views. And so we named what we want to do as something like the next generation fluorescence microscope. So in the end, I want to say that because my background is not biology, I'm totally a microscopist, also optical engineering. So perhaps you guys can teach me how to think as a biologist, and hopefully we can collaborate more. Not only during Janelia, but also among Janelia and EMBL, something like that. Yeah, and hopefully my microscope can be finished this year, or at least maybe next year, and we will have something fun. So here is all the talk I give today. Thanks for listening, and yeah. Question and answer time.
You mentioned something about the time scale for being soon.
Time scales. So one phase is usually 10 milliseconds to 20 milliseconds. And you have 5 phases per orientation, which means that, let's say 20 milliseconds, 5 phases means 100 milliseconds. And you have 3 orientations, which means that 300 milliseconds, just one slice. And depending on your thickness of the sample, if you want to acquire, let's say 4 micron, then you need to take like 67 these steps. So you will have 67 multiplied by this 300. So each one takes about 10 seconds each step.
So do you think your resolution could be affected by movement, presumably biological, whatever is labeled? So this everything's moving in the cell.
Right.
At some size.
At some, yeah.
Scale. So
Exactly.
do you have any sense of whether, if you had gone faster, you might have captured better resolution?
Exactly. So that is also the reason why so many labs, they only focus about 2D thing or curve thing, right? You only need to acquire 9 images per plane. That is per plane is per stack. That is also per time point, which means that they can acquire like let's say 100 milliseconds. Then you can image the sample very, very quickly at 10 Hz. But if you go something from 2D to 3D, time scale will be extended based on how thick you want to image.
Thank you for a nice talk.
Thanks.
So you're trying to image deeper also, right? You're trying to image deeper in some cases. I'm trying to image deeper, like greater than 50 microns and so.
Can I say again?
So in your studies, you're also trying to image deeper into organisms, like greater than 50 microns and all.
Oh, you mean the imaging depth? You mean the work I'm doing now, or the 3D sim or being same?
So I'm just wondering how reliable is to estimate the number of products like post processing, estimated.
That is a good question, but I have to say that it's something out of my scope because I'm not so familiar with the pattern recognition or the fiber differentiation, something like that. So unfortunately, I cannot give you a very good question for this answer for this question. But I think we can discuss later. So because in our lab, Pluto in our lab, so he's quite familiar with kind of the fiber and differentiation, something like that, using the movie models. I think he's quite familiar with this part. Yeah, we can discuss later. It's a good question. Yeah.
So with 4D sim, you get roughly 160 nanometers, you said, right?
Yeah.
And have you tried doing the 3D arcane prediction on top of that? Does that help? Because you said you use it for denoising, but can you use it for resolution enhancement as well?
3D arcane itself, actually, the denoising model I used is based on 3D arcane. And the actual resolution model, there is no restriction to which kind of deep learning model you want to use. You can use 3D arcane, you can use CARE, you can use whatever other density convolution net, you can use regionalized deep learning. So here the concept is not, so I can show you the. Oops. So here the concept is not which kind of deep learning model you want to apply on to increase the actual resolution, but to all your data sets should be processed along the axis view, and you digitally rotate it, because you can blur the axis. Axis view somehow it is blurred along Z, but X is still quite fine, right? Because X is is still super resolution. Then you can blur the X direction to make it isotropic. Then you can build the neural network from this isotropic but blurred XZ view to the original fine features which has super resolution along X direction. Then so for this neural network establishment, I actually don't care which kind of model I'm using. So we can use whatever one you want to use. But the key point is that you have this model and you digitally rotate it by 6 directions along the XZ view. And you will have one, always have the resolution improvement prediction along the X direction, but you have to know that this X direction has been rotated. So you need to rotate it back. And you merge these rotated images, then you can get the isotropic resolution along the XZ view. That is the key point here. And the Y view, you did nothing on it. So it's always super resolution. Right? So you have isotropic. That is somehow stuck here. Anyway, yeah, something like that. Hope I can explain it clearly enough.
Sorry, Robert, you have a question. I missed you there. Can you unmute yourself?
Sure, no worries. I was listening. Yeah, thanks a lot. It was a really exciting presentation. I think I have a question.
Great,
the former part, right, where you compared the 4D sim and 3D sim plus sort of image reconstruction and denoising. And my takeaway point, the takeaway point that I got was that the later one is better, right, because it says photo bleaching. But the problem is also in the validation part. So have you thought about a little bit of a more clever hybrid approach where you just use the 4D sim to validate some of this AI denoising and reconstructions? Sort of where you would not sort of acquire a 4D sim all the time, but you would do it, I don't know, once every while, because once you sort of image your sample in 4D sim, you can use that information to train and validate the 3D sim plus AI reconstructions.
It's a good question. Actually, some other people has also asked me the similar question. The answer is actually you must, right? For deep learning, there is no ground truth because you improved the actual resolution. And how do you know that your deep learning prediction, whatever method is correct, right? It is really genuine, is accurate.
Exactly.
I have to say that for one structure, I would not say it's one structure. Let's say if you want to image some microtubule-like or actin-like structures, you just use the four beam sim to get the near-isotropic resolution improvement just once, right? And acquire that sample using 3D sim plus deep learning to get the prediction. Then you compare these two results. If you think, okay, it's close, then we trust the deep learning prediction. But I have to say that you cannot do all the deep learning and all the four beam sim all the time. It's really time consuming, right? But for one structure, you do the validation, you think that it's close or correct, then you should be safe to use this imaging model. And of course, this actual resolution modality is not almost 100% accurate. So what all the movies I show here is after several rounds of validation from the four beam sim, we are quite confident that this structure should look like this along the axis view, and we put it here. But also some other features which perhaps is too close to each other, right? Let's say it's like sub 100 or even like 20 nanometers, right? Then whatever four beam sim or deep learning, they cannot give you some reasonable results. And even these two results, they could be quite different from each other. Then we will say, okay, this kind of structure or some organelles, right? They are not so suitable to use this actual resolution enhancement model.
Yeah, no, exactly. Maybe just a very quick follow-up question because I've been sort of brainstorming a little bit. So could you somehow sort of switch on and off the mirror? Is there a nice way to kind of block the mirror to do the 4D sim, to like a kind of toggle in between 3D sim plus and 4D sim?
Difficult. So there is not a really fast switch mechanism in my current setup. But hopefully, if someone follows this project, I think they can find some smarter way to do it. But currently, no. Yeah.
But yeah, thanks a lot for your really nice presentation.
Yeah, thanks.
If you have a 3D sim, how easy is it to add that mirror? Or if you had a 3D sim, how easy is it to upgrade?
So it's something like that. I did not put the 3D sim setup, but it's just the home build 3D sim setup. You have SLM, you have the liquid crystal polarization rotator. You are quite familiar with all the triggering. You build a 3D sim, then you add this mirror. So adding the mirror is simple. Align is not that easy. You need much of experiments with it. And I have to say that we are not the first group to increase the actual resolution on 3D sim. And Matt, in his 2018 paper, the first 3D sim paper, Lin Shaw and Matt, they published a back-to-back paper So they use a second objective on the top and do the 6-beam interference. That is totally a nightmare, I can tell you. So my setup, I have to say, is not easy, but it depends on who you compare with, right? So there is also a relatively complex phase maintenance or say feedback mechanism to maintain the actual phase stable. That is inevitable because phase is always stable along the XY direction. But the thermal drifting, any vibration could make your interference pattern somehow not correct. So if you have a correct phase or say constant phase, then you will have all the correct the small beats, like if you plot the line, it will be 160 nanometers. But if it's not correct or totally wrong, you have a double ring artifacts. We know what will happen, so you must be careful to to build this system. But I have to say, it's just not easy. I won't say it's difficult.